Hello, and welcome to the GRACE podcast series. My name is Denise Brock, and I am the Operations Director for the Global Resource for Advancing Cancer Education, or GRACE. In this podcast series, we interview patients, advocates, and healthcare professionals to provide the most updated information for our community and to highlight important issues facing those dealing with a cancer diagnosis. We hope you find this information valuable. For questions or comments, please visit us at cancergrace.org.
Hi, I'm Dr. Jack West. I'm Associate Clinical Professor in Medical Oncology at the City of Hope Comprehensive Cancer Center and also the founder and president of GRACE, Global Resource for Advancing Cancer Education. I'm very happy to be joined today for an ASCO highlights presentation in the field of lung cancer with two of my friends and colleagues from other parts of the country who are lung cancer experts with some different perspectives, and we're going to go through some of the key presentations and talk about what we think this means for patients. First, I have Dr. Helena Yu, who is medical oncologist at Memorial Sloan Kettering Cancer Center, and Dr. David Spigel, who is Chief Scientific Officer and Director of the Lung Cancer Program at the Sarah Cannon Cancer Center in Nashville, Tennessee. Thanks, guys, for joining. It's been a very busy spring in 2020 in terms of new FDA approvals, and one of them was for a new agent against the target of MET, specifically MET exon 14 mutations, which are in about 2 to 4% of patients with non-small cell lung cancer. This agent, Capmatinib, it now has the marketed name Tabrecta, and we knew it had good activity. We saw a little more data at ASCO 2020, looking at cohorts of patients who got it either as a second or third line treatment, and a smaller group on the right in the orange table who got it as first line treatment. And this is just more information that tells us what we already concluded, which is that this is a very active drug for patients with this target if you identify it. You can also see that the response rates are clearly higher if these agents are given in the first line setting versus later. And so this is the data with Capmatinib, and you can also see that it induces good deep responses, and you can see part of the brain MRI of one patient, just really highlighting that many of these targeted therapies have good activity in the brain, both treating brain metastases that exist and also in usually or hopefully preventing new brain metastases from developing in these patients. I put, it's a different target, but a similar category. The drug Pralsetinib is from Blueprint Medicines, known as BLU-667 as well. This also had new data presented by Justin Gaynor from Mass General, and we saw very good responses for patients with a RET fusion. This agent is not FDA approved, but there is another agent in this category targeting RET fusion called Selpercatinib with very similar data. And here you can see response rates of about 60% in nearly every patient with at least disease control. And then shrinkage of brain metastases here on the right, on the left, you can see a so-called spider plot where downward trend over from left to right represents shrinking tumor. And there weren't a lot of patients to speak to, but 7 of the 9 patients who had measurable brain metastases had tumor shrinkage on this. And so these agents are now FDA, increasingly FDA approved. Pralsetinib hopefully soon, but we already have another one for RET fusion. And it's exciting work. It's a new opportunity for a small population. I would say one of the challenges, though, is that you're never going to find it if you don't go looking for it. This is not, and these are the 6th, 7th targets in a list. And so I think that the only way that people are going to find these is if they do next generation sequencing, broad molecular testing, not one by one testing of a couple or 3 choice targets. Helena, can you talk about, a lot of this work's been done actually at Memorial. So can you talk about how transformative this is, or how limiting it is if, in terms of the challenges of next generation sequencing not being done necessarily in a timely way on a broad setting yet?
Yeah, I mean, I think that, as you mentioned, Jack, I think as we're almost in the double digits with FDA approved drugs for these different targets. You'll never find something if you don't look for it. And I think if at all possible, I think with these really sort of incredible response rates and durations of response, if we can give these first line, we really should try to give them first line. As we know, with metastatic disease, some people don't get to second line and later treatment. So it's just so important to use the best treatment first, and so I think, the faster we get with next generation sequencing, if we can, have the results within 2 weeks, or if they get sent off earlier, that will be really helpful for our patients.
David, anything to add? Or you are working kind of on the interface. You have a pretty academic practice doing a ton of research, but you also work in a broader, community setting for a lot of this research. How much or how little of a barrier is this need for next-gen sequencing to find these?
Well, I mean, not much to add, because I think the stories are so compelling. These drugs are so effective, so active. You go back to TREC even. I think community oncologists are recognizing that you're only going to find these if you test broadly, and you just kind of get into a habit of doing it. And, it's not just this, right? I mean, there's TMB just got to be an approved strategy to use immunotherapy, last week. We talked about HER2. We didn't even talk about RAF and ROS and ALK and EGFR to any great extent, but there's so many things we need to look for. Broadly testing makes sense. And so what we're seeing in our network are physicians talking more with their patients about this, and then trying to find the best way to do that for them. And often that ends up being either sending tissue off to an outside laboratory to do that analysis, or blood to get back at least a snapshot of some of those potential genetic alterations. So the main thing is, can you move somebody from not doing any testing to testing? And the answer is yes, it's happening every day now. And then trying to help them understand what those results mean, and then how to use that to apply to a treatment. This kind of discovery, let's say you're a patient tomorrow, a doctor tomorrow with a patient, that might be your only patient with this kind of alteration for a year or two in the community. And so having experience is not going to be easy for physicians. So this is going to be something that is going to take a lot of education and communication.
But I also think that, fortunately, it's a setting where, if you have a patient who you're doing NGS on everybody, if you find someone who's MET positive and you give Capmatinib and get a very good sustained response, that not only is going to inspire you to keep looking for MET, but it'll cross over to the same concept applies for RET and TREC and everything else. It's just, it's the same process and the same both testing process and thought process that applies from EGFR and ALK and ROS that we've been doing for a decade or more to these newer ones. It just, once you get beyond 5 and certainly approaching 10 or more targets, it only makes it more screamingly compelling. And I think now it's fully, the scales have fully tipped to one side, and hopefully at this point it will be compelling enough that just about everyone will decide we need to do whatever it takes to get this broad testing done.
Thank you again for joining us. This podcast was made possible by the generosity of sponsorship from our friends at Lilly, Novartis, Takeda, AstraZeneca, and Exelixis. Please like and follow us on Facebook and Twitter. Send us feedback, share your story, donate and visit us for more information at cancergrace.org. Thank you for listening.